before, and I showed it as an equation in the last class. This time I'm showing it to you as a diagram where you can see the enzyme that has the active site to where sucrose binds, and then the reaction occurs in the presence of water, which is hydrolysis. And then we have the products, glucose and fructose. Okay? Now, this is another value I didn't give you before. The reaction of hydrolysis of sucrose is actually gives you a minus 7 kilocalorie per mole delta G value, which means the reaction is spontaneous, right? But we explained in our previous slide that we can have sugar on our table, and it's not going to be hydrolyzing for the same reason, because it doesn't have the energy of activation to do so. Okay? So if you dissolve sucrose in water, again, we're not seeing hydrolysis either. We're just seeing the hydration shell solubilizing the sugar. But if you add the enzyme, if you add sucrase to that, then the sucrose would hydrolyze very quickly into glucose and fructose. Okay? So this is what we're talking about, the activation energy, the little pump of energy that spontaneous reactions need to start and continue all the way down to the product. And we also mentioned in our last class that what enzymes do, the only thing that they do is that they lower the amount of energy needed or required in here, in this little pump, the energy of activation, for that reaction to proceed. Okay? And we're actually going to talk about how the enzymes do this. So let's see if you can watch this little animation here. Okay. So, I'm going to start making the talks here. So we said that the reactant or the molecule that is going to undergo the reaction is the substrate. And what's going to help it speed up the rate or lower the activation energy is the enzyme. Okay? So we're going to show you a lot of diagrams, but this is more fun. This is what you would see an enzyme is like in your book, right? Represented as a molecule. And we know that enzymes are made up of what? Proteins, okay? So polypeptide chains. And you can see that within the protein structure, we have secondary structure, tertiary structure going on there. We have the alpha helices and beta chains and all that stuff that are making the molecule. And most enzymes, as I mentioned earlier, are made up of several subunits. So we have that the active enzyme can be 2 or 3 or 4 polypeptide chains working together. So when the substrate binds to the enzyme, that's what we refer to as the enzyme-substrate complex. Okay? And the enzyme is very, very specific for the substrates that it binds. So it will only bind what fits in the active site. Okay? So the active site is precisely where it actually binds to the substrate. Now, the enzyme may have a slightly more open structure, and once the substrate binds to it, then it kind of closes a little bit to make it fit better. And that is the model that we refer to as an induced fit. Okay? So once the molecule or the substrate has bound the active site, the enzyme kind of hugs it a little bit. Okay? And that gives it the conditions that are required for the reaction to proceed. Okay? All right. Let's see what's next. Okay. So we said that the active site is actually where the substrate, where the reaction occurs, basically. Okay? So we said that this is made of polypeptides, right? So the active site may have only a few amino acids that are very, very important to provide a microenvironment that is very specific for the reaction to occur. Okay? So we said that it kind of makes that energy push to be smaller. And remember that analogy of putting molecules in a pool versus putting them in a cup? So it's kind of the same. You're restricting the conditions of the reaction to this little spot right here. Okay? So once the substrate binds the enzyme, the reaction occurs, and we end up with the products. Okay? And the way that the enzyme helps that happening is because, first of all, if we have 2 molecules that are the reactants that are going to be part of the reaction, it can help by putting them in the right position. Okay? Facing each other in the right position for the reaction to occur. That's one way. Another way that it can happen is if it actually, once the substrate fits into the site, when it hugs it, it kind of strains the bonds within the molecule that is the substrate. So since those bonds are now under strain, it's easier for the reaction to progress, so to speak. Providing a favorable microenvironment would be, for example, well, we know that there are a lot of different kinds of amino acids, right? We have basic amino acids, acidic amino acids, with nonpolar chains or polar chains, right? So let's say this particular substrate needs acidic environment, so it can hydrolyze. And we are in a neutral cell, cytoplasm. So what happens is once the substrate binds here, guess what kinds of amino acids are here? Guess. Amino acids that have acidic side chains, right? So they are creating this acidic pocket that allows the reaction to occur properly, okay? Also, another way that it can happen is if the enzyme covalently bonds to the substrate for a little bit of time. It covalently bonds to the substrate, and that causes a change in the shape of the substrate itself, and then that promotes the reaction to move forward. Okay? But again, those would be transient bonds. Okay? It's not going to stay stuck there forever. So, explained in the diagram, very nice here in the screen. You have the substrates, in this case these are two substrates that interact with each other, that bind perfectly to the active site of the enzyme, creating the enzyme-substrate complex. Then we have the reaction happening, okay? And the substrates are transformed into the products, which are then released from the enzyme. Okay? And then the enzyme is free and unharmed and unchanged to go again and do the same thing all over again. Okay? And we're seeing this as an example of a single enzyme working, but these type of reactions happen, all the time. They happen thousands of times in a second or so forth. And they have a lot of these reactions happening, one after the other. So we don't have a single enzyme doing this. We have lots of enzymes doing this at the same time. Okay? All right. So an enzyme to work properly, it needs to be in the right environment. Okay? We said that form fits function. And a lot of the environments, changes in the environment may cause a change in the shape of the protein. And if we have a change in shape, then it may not work as well or at all. Okay? So we have environmental conditions such as temperature and pH that directly affect the activity of an enzyme. So a particular enzyme may have an optimal temperature to work. That is, it could probably work a little bit less at a lower or at a slightly higher temperature than its optimal, right? But it's the best performance happens at a very specific temperature. The same thing happens with the pH. We have enzymes that work at different pHs, okay? And the optimal pH, of course, has to do with where that enzyme needs to work. Okay? So we're going to explain that in advance after you've answered this question. Okay, how are you all doing? Are you good? I forgot to start the timer, sorry. So, if you have your answer, go ahead and submit. I'm going to stop the question. 1, 2, 3. Where's my graph? Technology, help me. I don't see my graph. Okay, let's answer the question, because I don't see the graph here. So we're talking about a common situation. You have acid indigestion, right? That you ate something and it didn't agree very well with your stomach. And one of the first things you do is you take Tums, or malose, right? Tums is calcium carbonate. And we know that this helps with the indigestion, okay? And we're telling you here in the question that it's partially because it inhibits the activity of the enzyme. That is, it prevents enhance the activity of a digestive enzyme. Okay? So, having said this, what do you think will be the best condition for that enzyme to work properly? So, for starters, where did we say we're having the discomfort? In the stomach. Okay? So, would it be relatively basic? No. Okay. Whoops, there's a graph. So, no, it's not relatively basic. Would it be relatively neutral? No. Okay, so it will definitely be relatively acidic. And it didn't show the check mark, but that's the right answer. Okay, so we have, as I mentioned earlier, that enzymes have a very, very specific temperature at which they work the best. And that's what optimal means. The best performance happens at that particular temperature. If we're talking about enzymes in the human body, we know that our temperature in degrees Celsius, our normal temperature should be around 37, or very close, okay? So if we're talking about enzymes that work in bacteria that live in the hot thermal vents in the bottom of the ocean, 37 centigrade is probably too cold for them. Yes? We're actually going to talk about that when we talk about homeostasis, but when you have a fever and your body temperature goes up, a lot of things happen in your body to try to regulate that, so your enzymes won't denature. So we said that the application of high heat may denature enzymes. What did we say denaturing is? What happens? When a protein unravels or unfolds, yes. So it changes shape, so it's not working. So we have that an optimal temperature for a typical human enzyme is 37, right? And if you go a little bit below 37 or a little bit above 37, it still works, okay? It works slightly less efficiently than it would work at 37, but it still works. So if you have a fever and you're 40 centigrades in your body, most of your enzymes are going to start to have a problem because any temperature above that will start killing them. Will start unraveling those, denaturing those proteins. So that's why we take care of fevers right away. And any temperature below that, below 37, the enzyme would work, but less efficiently until, of course, you reach 0, it doesn't work, 0 centigrades. So in the case of bacteria that are thermophilic, that means that they love heat, we have the thermal geysers in the national parks or the hot thermal vents in the bottom of the ocean. There are a lot of prokaryotes that live there, and they actually adapted to those environments by having enzymes that have their optimal activities at high temperatures, right? So if you get an enzyme from a bacterium that has an optimal temperature of 77 centigrades, and bring the temperature down, then you won't have the same activity or the same efficiency of activity of that protein. Same thing happens with pH. Okay? We have enzymes that work or that we need them to work in the cytoplasm of the cells. And we said that the cytoplasm of the cells is very close to neutral. Okay? So they should be working around here. But if we're talking about digestion, like pepsin that we've talked about in the previous question, pepsin works in the stomach, and we have a pH of 2 there. Okay? So that's the optimal pH for that enzyme. If you change the pH, there are other type of things that we take when we have acidity or indigestion. Maalox, I think. Maalox will bring up the pH of the stomach. Okay? and if it brings up the pH of the stomach slightly to 3, so to speak, then the enzyme is not working as efficiently as well. Okay? And then we have trypsin. Trypsin is an intestinal enzyme. And the intestines have a completely different pH than the stomach, as you can see. It's basic. Okay? So for trypsin, a pH of 8 is optimal. What will happen to trypsin if you put it in the stomach? What happens? It's not going to work on one hand. On the other hand, it's going to unravel, right? Nature. So if you take it out of this range where it can still work, here it's denatured, here it's denatured, and so forth. Okay? Any questions? Okay. I think this slide wasn't previously in your notes, so it's important that you write information here. Because the rate at which an enzymatic reaction will proceed has a lot to do with the amount of substrate and in some cases with the amount of enzyme. So they kind of go hand in hand. When you have low substrate concentrations and the enzyme is working fine, if you add more substrate, then you can speed up the reaction. But there's always a limit to how fast the reaction can occur. Okay? In the opposite case, when you have a lot of the substrate, then what happens is that if you have a certain amount of enzyme, it will all become involved in the reaction, and it will become what we call saturated. Okay? So there are no more available active sites of enzymes for the reaction to keep occurring. In that case, the reaction is going to slow down, the rate of reaction is going to slow down because, of course, all of the enzymes is engaged. In that case, what you could do to speed up the reaction is add more enzyme. Okay? Now, what's going on there? All right. So, what happens in biochemical pathways when we're talking about metabolism? All those metabolism, anabolism reactions that we started mentioning in the last class. The enzymes that are involved in the reactions sometimes work in the direction of breaking down the reactants into products, and sometimes the same enzyme works in the opposite direction. Okay? And at that point, in which direction are the enzymes working, through the breakdown or the opposite, it has to do with the amount of reactants and products. Because all reactions tend to what? Equilibrium. Exactly. So everything wants to be as stable as possible. So we want to reach equilibrium. Though we never want to reach equilibrium, right? So the reactions are moving in the direction towards equilibrium. So, okay, enzymes are very good at doing what they do, but sometimes enzymes need help from another type of molecule. And as you will see in a little bit, these molecules are not just there to help, but they're also to help us fine-tune pathways. Okay? And if these molecules that help the enzymes are inorganic or non-protein, we refer to them as cofactors. So they can be inorganic or organic. Okay? So can anybody think of inorganic cofactors or metals that make up enzymes? Zinc is one of them. We have zinc. We have iron. We have copper. Okay? And if the molecule that is helping an enzyme is organic, of organic origin, then we call it a coenzyme. So So coenzymes include all the vitamins that you ingest in your food. Vitamins are important to you, but did you know why? Because they help your enzymes to work properly. Okay? So we have the molecules that help enzymatic activity, and then we have molecules that halt or stop enzymatic activity. And these are called inhibitors. Okay? We have different kinds of inhibitors, and again, inhibitors can be regularly used by living organisms to fine-tune metabolic pathways. Okay? We have different kinds of inhibitors. We have the competitive kind, and they're competitive because they actually compete for the active site of the enzyme. So remember the enzyme? We have inhibitors that are actually very similar in one side to the substrate, so they fit into the active site. And they fit into the active site and then prevent the substrate from binding the enzyme to the substrate site. Okay? So this can be semi-permanent or a temporary interaction that will be relieved. Okay? And now the enzyme is ready to do whatever else. Or this can be like a permanent type of modification in which the inhibitor remains attached to the enzyme on the active site. Okay, let's see. Let me go forward one slide and then I'll come back. So there are non-specific type of inhibitors which what they do is actually cause the denaturation of the enzyme. Right? And we talked about acids, bases, temperature, other substances like in the case of alcohol, heavy metals, or reducing agents. Those cause the unfolding of the protein, and then the enzyme is not active. But then we have the specific kind of inhibitors, and they can be irreversible, okay, when the inhibitor is permanently bound to the enzyme and that enzyme cannot go back to its active state. Or they can be reversible, that is a temporary interaction that then is relieved. And it can be competitive, like I showed you. And it can be non-competitive or allosteric. And then have also feedback inhibition, which is allosteric as well, as you will see. So let's go back. Okay. I wanted to, I'm not sure if I missed something from the previous slide, but okay. Thank you. Okay. So we have the enzyme, and we have the competitive inhibitor binding here. Okay? And then we have the non-competitive kind, which is actually a molecule that binds on a different site in the enzyme. Do you see that this one has a different shade? It has a different site there. And what happens is when the molecule binds here, then the enzyme changes shape. And when the enzyme changes shape, then it doesn't fit the substrate anymore. Okay? So if this is a temporary interaction, when this one goes away, the enzyme will kind of fold back to its regular shape. Okay? But if this is an interaction that happens, that sticks, so to speak, then the enzyme remains inactive. Okay? So the same is shown on the slide behind me. We have the substrate binding to